So the last speaker for this session is Fabrice Touzin. So Fabrice is a researcher at the French Agency for Food, Environmental and Occupational Health and Security and Safety, ANSES, in France. So he works at the laboratory of Ploufragan-Plouzané in viral genetics and biosecurity unit. So Fabrice, since 2012, is a researcher in this unit. He is the team leader for the Ploufragan bioinformatics team, and he works in production of treatment pipelines for high-throughput sequencing data like Iron Torrent, Illumina, PacBio, and Minayen, related to different organisms like emerging viruses, bacteria, plasmid and antibiotic resistance, mitochondria, and finally for the detection of transgenes in undescribed genetically modified bacteria, which will be the subject of this presentation. Go ahead.
Thank you for the introduction. I would like first to thank the organizer to have invited me to present the work of Julie Aurel, the PhD student I supervised for the bioinformatics part. She worked on the detection and characterization of undescribed genetically modified bacteria by statistical analysis of high-throughput sequencing data. So for the machine learning part, she was supervised by Stéphanie Boujard. So this work was done in the viral genetics and biosecurity unit at the ANSES of Ploufragan in Brittany, France. So let's take a look on what we will treat as a GMO in our application. It's a draft, kind of. So I will call host genome the wild-type genome in which you can introduce one or several inserts, CDS, GM-CDS. To detect GMO, you've got several methods. For non-GMOs, you've got a method based on protein detection and DNA detection, among which qPCR, you've got GMO-seq. For partially known GMO, you can use new sequencing techniques coupled with molecular methods, bioinformatics statistics, and you can use DNA walking. But for unknown GMO, there was no method available so far. So here is a summary of what we can do for the detection. So as soon as you know the GMO you are looking for, you can use PCR, and it's quite easy to do. But when you don't know at all what is in your GM sample, you have only high-throughput sequencing and deep analysis by bioinformaticians and specialists in the field. So our objective was to develop DUGMO, a detection method for unknown genetically modified organisms. The basic idea is to identify the vocabulary differences between the host genome and the insert. So we use the CDS of the wild-type genome, so host genome, to determine a specific genomic vocabulary that is species-specific and composed of words. For instance, if you take a species 1 and you consider 4-letter words, you will have overrepresented words, 4-letter words. And if you take a species 2, you won't have the same words that will be overrepresented. So if you take several word lengths and Markov model, you can define the vocabulary used by your species. So here are the main steps of DUGMO. Data preparation first, then characterization of the host genome CDS based on their overrepresented words. Then the calculation of the distances to support the comparison to the host genome CDS, and the design of the prediction model. So after data preparation in your sample, you will have several data sets. The set of the CDS of the whole genome, so wild-type CDS, they characterize what we are not looking for. This is the most important data set, in fact. Still, in your sample, you have the set of potential GM-CDS. They are absent from the pangenome. You want to evaluate for each of these CDS if it's a GM-CDS or not. And finally, you've got a data bank of non-GMO CDS. They characterize the non-species sequence viability, and they are not in your sample, of course. You don't know. So after that, you compute the distances to characterize the vocabulary of A, so Y-type CDS, the vocabulary of C, the GM CDS, and finally you want to deduce the vocabulary of each sequence in the set B, so as to determine with your prediction model if each CDS is a GM CDS or not. So here's an overview of the DOGMO tool. So you've got preparation of CDS, extraction of 9 variables of interest, prediction model, and then results. First, recall on what is the pangenome. So a pangenome is several genomes of the same species that represent the variety of all the genes of the species, or almost all the genes. In our case, we want that the pangenome is a Y-type pangenome, and that is representative of the variety of the genes. So in the input of the method, you've got high-throughput sequencing data of the sample you want to analyze. You've got a reference genome, a Y-type reference genome, a Y-type pangenome of the same species, and its associated CDSs. And finally, you've got a data bank of known GMO insert. So we will clean data, assembly, annotate, sort, and from our high-throughput sequencing data, we want to know for each CDS if it's related to the genome, Y-type CDS, or if it's a potential GM CDS. So first step is cleaning. From the read, we do an assembly using Shovel to obtain context or scaffold. We use Proga to do annotation and have CDS of your sample. Then we compare our CDS to Y-type pangenome CDS. If they are similar, it means that the CDS of our sample are Y-type CDS. Otherwise, they are still in the category of potential GM insert. Let's take a look more precisely on the comparison. We do a blast on the Y-type pangenome, and if we have an identity percentage higher or equal to 98%, or higher or equal to 95% with HSP percentage of identity higher than 98%, then it is a Y-type CDS. Otherwise, it is still sorted as GM insert. The next step is to clean the GMO data bank, because we have to remove from this data bank everything that is similar to the species we are analyzing, Y-type species. There is a second step to remove the CDS that corresponds to the same vocabulary than the one of our Y-type species. And finally, we have 3 datasets, each CDS of known GMs and GMO. And in our sample, each Y-type CDS, and finally, each potential GM CDS. Now we need to compute the distances. Our objective is to characterize the whole genome CDS and the CDS of the 2 other datasets. For this purpose, we determine the best combinations of parameters to establish explanatory variables for the development of the prediction model. For the characterization of the genomic vocabulary, we use RMS that search for exceptional words in a sequence. It determines the number of occurrences of each word and its exceptional character. And it defines a set of words that are overrepresented in the whole genome. We tested several distances, Euclidean distance, Kullback-Leibler distance, and finally Berry-Curtis distance, that is more dissimilarity measurement than a distance. And we computed 2 types of distances. The first one was called infrequencies. So we took the 27-letter words in our CDS and we concatenate the third position of the codons of this CDS so as to make it computable by RMS. Otherwise, the 27-letter words are too long to be analyzed by RMS. And we returned 10% of our represented words in Y-type CDS. Why is the third position of the codon? Because this is the position where you've got the selective pressure that is the most related to the codon usage of the species, and so to the species. And we computed another kind of distance that we called in proportions with the whole CDS and all words, 3 and 4 letter words. We tested a high number of combinations of parameters, among which word sizes, order of Markov model, percentage of over or underrepresented words, concatenation of set positions of codon. And finally, we retain Brackus distances in frequencies with 9-letter words and a Markov model of order 7, but this 9-letter words corresponds to 27-letter words in your CDS. And distance in proportion with 3 and 4-letter words with Markov model of order 1 and 2 respectively. We also retain the average exceptionality score provided by MS in the whole genome for L4M2 and L9M7. The count density per nucleotide for 4-letter and 9-letter words, the percentage of GC, and finally the length of the CDS. Then we can compute the distances. We compare all the time to all the CDS of the wild-type genome. And we compare to each CDS of non-GM CDS to obtain 3 Brackus distances and 6 other variables and a vector associated. And we do the same for each CDS of the whole genome, so wild-type CDS, and finally each CDS that are potential GM in our sample. Each time we get vector that will be used for our prediction model. So we have 2 datasets for the training, non-GM CDS and wild-type CDS, and we want to predict in our sample for each potential GM CDS if it's a GM CDS or a wild-type CDS. Julie tested 12 different methods, either parametric or non-parametric. So we have in input 2 training data sets and 1 predictive data set. We use centered, reduced, and stratified data. The optimization of the parameter of each method were done in 10-fold cross-validation, and we use 2-fold cross-validation comparison of methods. Our selection criteria were false negative rates as low as possible. This is the most important criteria. Then the specificity and sensitivity, and finally the false positive rate. We retained the union of the result of the random forest machine learning method and the logit method for generalized linear model to obtain a false negative rate of 1%, a specificity of 99%, a sensitivity of 99%, and a false positive rate of 9%. So we tested the method on 4 GM bacteria, on wild-type bacteria, and a lot of GM synthetic bacteria. And finally, we found almost all the GM CDS except 1. And in the wild-type CDS, we didn't find GM CDS except 2. So we have 2 false positives. The first one can be explained by the sequencing data, because when you do sequencing, if you have a coverage depth that is quite low, you will have sometimes genes that can be truncated. In this case, truncated genes are detected by DUGMO as GM CDS. So the solution for this is to be sure that your sample has at least a coverage depth of 60, or even better 100. Sorry, as here. The second false positive was, in fact, a gene with a low identity percentage, just under the cutoff, and we Z4 into the NCBI data bank in all the actinomy sets and didn't find any other match in actinomy sets. It was in Mycobacterium tuberculosis, and we think this is an horizontal gene transfer. It is one of the applications of DUGMO also. Finally, Julie tested the sensitivity of the method to decodon optimization, codon and decodon optimization. She tried a lot of times, and in all cases, DOGMO was able to distinguish them. She tested the detection threshold of the method. For this purpose, she introduced substitution in a wild-type CDS until we detect it, and DOGMO is able to detect modified CDS if he has at least a substitution rate of 9%. In conclusion, here is what we had before DOGMO. Now we can tell that we can find this kind of unknown GMO for bacteria, and pure bacteria, of course. In the slide, I forget to mention the GM that corresponds to the suppression of a gene. Of course, DOGMO won't find it because it's not in the sample. And we expect to extend the method to eukaryotes. This is one of our perspectives. And we will give the possibility to provide assembly data as input from Illumina or PacBio sequencing. Finally, I would like to thank Stéphanie Boujard for the machine learning method. It was a big part. Vincent Ayrault, Mathieu Roland, and Anne-Laure Boutigny for the biological questions. And a huge thank to Sophie Jbat for the statistical, for the discussion on the world statistics. And thank you to Mauro Puterio, Frédéric Debonne, and Lutz Grumman to have provided some GM sequences. And finally, a good thanks to our funders, mainly the Brittany regions, so Région Bretagne. I thank you for your attention. If you have any question, I will be pleased to answer.